I'm Aaron from Lowell Team A.
Hi, I'm Candice Ng from Lowell Team A.
I'm Flora from Lowell Team A.
I'm Savini from Lowell Team A.
Our project focuses on a special protein found in malaria called RVAR2 that allows Plasmodium parasite to locate and identify cells that have a CS or chondroitin sulfate carbohydrate chain. Not only is this CS chain found in placenta tissue, it is also related to cancer cells because the CS chain is also found in the sugars of cancer cells. So our project focuses on how we can use the RVAR2 protein to stop the uncontrolled cell proliferation of cancer cells.
Cancer is a disease caused by an uncontrolled division of abnormal cells in a part of the body. Cancer has been associated with death since it was first diagnosed and researched. Anyone from all walks of life is susceptible to cancer. In our project, we are specifically targeting prostate cancer, breast, lymphoma, and melanoma cancer, and also gastric cancer. Fortunately, today's advancing technology in the medical field provides large glimpses of hope to finding a cure to cancer. RVAR2, the recombinant protein with chondroitin sulfate, is produced from the deadliest malaria parasite. It is the key to an alternative to traditional cancer treatments. Chondroitin sulfate, which consists of a glycosaminoglycan glycan sugar chain, is found in the placenta of pregnant women and has been discovered recently in various tumor cells and tumor stromas. RVAR2 permits the adherence of the parasite to the host red blood cell through binding with the chondroitin sulfate. As the placental type CS is also found in tumor cells, the RVAR2 protein is able to serve as a tumor locator in the body. Binding the RVAR2 protein itself to tumor cells has shown to inhibit cell interactions, decrease cell migration, invasion, and encourage independence growth of tumor cells. We can then fuse RVAR2 with an immunotoxin. The specific toxin we will use is the diphtheria toxin, which is activated by proteolysis, the breakdown of protein into smaller amino acids. After proteolysis, two polypeptides are produced, a catalytic C domain and an R receptor binding, and T transmembrane domains. The R domain also assists in binding the toxin to cell surface receptors to trigger receptor-mediated endocytosis. The T domain mediates the transfer of the C domain into the cell. C-terminal domain facilitates receptor-specific binding to cells and blocks protein synthesis through the inactivation of elongation factor 2, which is necessary for protein production. However, the C-terminal binding domain can be replaced with an alternative ligand that binds the RVAR2 protein, generating a fusion protein that combines the ability of the RVAR2 protein to accurately locate the cancerous cells and the ability of the diphtheria toxins to kill cancerous cells. More specifically, RVAR2 on the fusion protein will locate the cancerous cells through detection of the CS triggers on the cell. Once the cell is located, the R domain will initiate binding of the protein to the receptors on the cell, resulting in receptor-mediated endocytosis. Subsequently, the T domain of the diphtheria toxin will deliver the fusion protein across the cell membrane and transfer the C domain into the cytosol. When the C domain will be able to inactivate elongation factor 2, which inhibits cellular protein synthesis and causing the cancerous cell to perform apoptosis.
So now for testing, benchmarkings, and comparisons. First, we like to test on lab mice, as testing on humans in our first trials would be too risky.